Good morning. I'm Dr. Dan Guerra, and this is another issue video of VerEvMed. Today, I want to talk to you about a particular form of a very aggressive breast cancer known as triple-negative breast cancer. It's one of these diseases that you hear about occasionally, both in the popular press and certainly in the medical journals, but it doesn't often get as much attention as does the other major form of breast cancer, which involves that associated with hormone sensitivity, particularly estrogen sensitivity. So we'll see why that's the case, because the prevalence of triple negative breast cancer is lower than the hormone sensitive type, but also it might have something to do with the complexity of it. So let's just get started with this talk. So we're going to talk about new pharmacological therapies for triple-negative breast cancer. Again, I'm Dr. Dan Guerra, and this is a VerEvMed lecture, and I'm giving it on the 17th of November, 2017. What is triple-negative breast cancer? First of all, it lacks expression of estrogen receptor, progesterone receptor, and the human epidermal growth factor receptor 2, or HER2. This involves an alteration of the expression of these genes, that is a diminution of them, as well as a lack of overexpression to compensate of the HER2, and also an alteration of gene amplification via copy number. So TNBC accounts for somewhere between 12 and 70% of breast cancers. Worldwide, it's probably up closer to 20%, but in the US, it's about 12 to 17 reported. Some things about this type of breast cancer. It is earlier age onset, so younger women get it, premenopausal that is. It's highly aggressive, and so it's basically a high-grade carcinoma. Along with other cancers, this particular cancer is even more enhanced in genetic instability and heterogeneity within the cell population of the tumor. So it's a high-risk early metastasis as well because of this aggressive phenotype. And death, unfortunately, can usually occur within 5 years post-diagnosis. So it's a very bad player, very bad disease, so we need to take a better look at it. So we know something about TNBCs. First of all, they're highly correlated with the BRCA1 germline mutation. Now, what BRCA is going to be a gene product that's involved in DNA repair. So I want you to keep that in mind. That's something that we have to carry out throughout the talk. So if you get a mutation in that, okay, then that's going to mean that there's something wrong with DNA repair. And that, in fact, is part of why this is such an aggressive tumor. So BRCA1 normally plays an essential role in DNA repair, also is involved in cell cycle checkpoint control and the maintenance of the genome stability. A mutant BRCA inhibition, so inhibiting the mutant, should annihilate DNA repair in triple-negative breast cancer, thus ultimately leading to apoptosis, okay, or programmed cell death, a topic I covered just recently in a VerEvMed lecture. So keep that in mind. If you have a mutant BRCA1, then if you inhibit the mutant form, you're going to annihilate any kind of DNA repair that might be afforded. So having a mutant BRCA means the DNA repair is corrupted. But for the tumor to maintain viability, you still have to have sufficient amount of DNA repair so that the cells constantly divide. With no DNA repair, all the cells would just go into apoptosis or perhaps senescence. So the idea is to further enhance, like pushing the BRCA1 mutation over the edge and annihilate it. That's one of the things we're going to see today and counted in this talk. So I told you that it was a germline transmitted disease. So you can have a carrier who's an autosomal dominant, a male, for example, that's not showing, not presenting with the disease. You can, if he is in the germline, he could parent, if he parented 4 offspring, 2 of them have a chance of carrying that allele because it's autosomal dominant. So that means that 50% chance of inheritance of that BRCA mutation, and that plays out to, because not all mutations lead to cancer, somewhere between 30 and 70% of those offspring could run the risk of BRCA associated triple negative breast cancer. So this slide over here shows you what BRCA normally does. You get DNA damage, or you get stalled DNA replication in the nucleus. A complex of proteins associate with FAN CD2, and that activates and monoubiquitinates, that's adding ubiquitin to this FAN CD2 protein. Then that induces BRCA1 from associated with an association to BRCA2 and RAD51. This complex here, BRCA1, BRCA2, and RAD51, is a DNA repair complex. And the kind of DNA repair it's involved in is that associated with homologous recombination. So it generates high fidelity. So obviously, if you have mutation in the BRCA1 or BRCA2, you're going to have a corruption of DNA repair. Now, in a cancer, that means that you can have runaway cell division, which then will lead to the oncogenic event of tumorigenesis. But again, if you push it over the edge and you get no DNA repair, okay, 0, then those cells will earmark themselves for apoptosis or programmed cell death. So there is a fine line over the brink of which the BRCA1 mutation can lead ultimately to the annihilation even of the tumor cell lineage. Okay, so I want you to keep that in mind. It's like that with all cancers, not just with the triple negative. All right, so when you have the triple negative phenotype, it has multiple subtypes. Okay, this is why the triple negative breast cancer is complicated, because there are multiple subtypes. So you can have the BRCA mutant, and you can also have the BRCA-NES mutant. You can have a mutant that's associated with the immune system. Okay, so that has to do with turning down or shutting off, for example, cytotoxic T cells. So there's another whole therapy that we can look at for that we talked in previous lectures about when we were discussing hepatocellular carcinoma, for example. There are also those that are called Claudin-low subtype. Now, what are Claudins? Those are tight junction membrane proteins, and they're normally expressed as junctions between the epithelia and the endothelia. And they form something called a paracellular barrier. And that paracellular barrier, along with the pores that are generated, so the junction actually occurs and that it's tight, allows for a very limited amount of permeability between the epithelia and endothelia. That's normally what Claudins do. So a Claudin-low subtype of triple negative breast cancer means that the expression of that protein is really low, which means you get not as much tight junction. Okay, and that always then causes problems because you get enhanced permeability. You see, enhanced permeability between cell masses usually leads to a dysfunctional communication, which can lead to all kinds of pathological events, including tumorigenesis. So get the idea, there's multiple subtypes. The two we're really going to look at today are the basal-like tumor and the Claudin-low subtype. Those are the two that we're going to be looking at today relative to the papers we're going to discuss. Now, just to give you an example of what it means when you are not triple negative, when you are estrogen receptor negative, okay, which is going to show on the slide here to the right, what happens when you have estrogen receptor positive breast cancer, okay? That would not be a triple negative. This would be one of the steroid hormone receptors are functional. This is much more common breast cancer. So when you have estrogen receptor positive breast cancer, the drug, for example, tamoxifen, inhibits the binding of estrogen to that receptor. And in so doing, you get a blocking of cell proliferation because cell proliferation is controlled by the estrogen in estrogen receptor sensitive breast cancers. Okay. You can inhibit that process with just taking the drug tamoxifen. Now, if you don't have an estrogen receptor, okay, you're still getting cell proliferation. This is going to be an autonomous replication, just not going to be associated with estrogen. It doesn't mean that people that have triple negative breast cancer don't have circulating estrogen. They usually do. It has very little to do with the accumulation of the hormone. It has to do with whether or not there are receptors for estrogen on the tumor subtype. That's the point we're talking about here. Likewise with the progesterone and with the HER2 receptors, lacking them on the tumor cells, not in the healthy host cells. Okay. So what does BRCA normally do? I told you that it was involved in DNA repair, something we talked about at length in a few of the other lectures I've given on this format. So you get DNA damage, which turns on a series of proteins called the ATM-ATRs, which then activates a protein called CHEK2. CHEK2 then causes the expression activation of BRCA1. BRCA1 blocks MYC transcription. MYC is often found as an oncogene. So BRCA1 blocks oncogene expression, but at the same time, it activates P53, which is, of course, a major checkpoint protein, right? In fact, it is a tumor surveillance protein, right? It's a tumor suppressor protein. And what it does when P53 is activated by BRCA via the DNA damage activation, you can get apoptosis. You can also get a series of checkpoint controls. That means control through the cell cycle. So BRCA1, I told you, also works through DNA repair mechanisms. So it's going to turn on this pathway here, the FAN-CD that we saw a few slides back, and in combination with BRCA2, RAD51 is going to be involved in DNA repair via homologous recombination, via that mechanism, okay? There's another pathway that BRCA works in terms of DNA repair, and that's non-homologous end joining. It's another mechanism. And then through these proteins here, you also get homologous recombination via BRCA1, and you also can get chromatin remodeling, which is going to afford an alteration of gene expression, leading to the overall global response of DNA repair. That's what BRCA normally does, right? It's very significant in DNA repair after damage. So obviously a mutation in that gene is going to corrupt repair, and it can lead to the lack of control of cell division after mutation, after DNA damage, and that can lead to an oncogenic event. So keep that in mind, bracket that in your mind, and now we're going to talk about the other major player in this talk. Now these proteins that we're going to be talking about have a kind of a nickname. They're called epigenetic readers, okay? So in epigenetics, just to refresh everyone's memory, what happens is that the DNA and the histones that are associated with DNA in eukaryotic chromatin in the nucleus become modified covalently, either via most common methylation of cytosine residues and CpG islands proximal to open reading frames, or acetylation, often found on lysine residues in the cohering basic proteins known as histones associated with the nucleosomes, where you can get active chromatin remodeling. The key feature here is that an epigenetic writer, that would be another component of epigenetics, will take a genome that has no epigenetic marking, which normally has transcriptional control via promoters and enhancer regions and transcription factors and single-stranded binding proteins. And that particular genome is going to be responsive to changes in DNA replication and changes in DNA damage and repair. Like this, in other words, is a healthy, non-epigenomically altered area of the DNA, okay? So a locus of the DNA. Now, as it turns out, a lot of eukaryotic DNA, that is human DNA in particular, is epigenetically written or rewritten. And so the writing is caused by a series of proteins, which can be the acetyltransferases. So you have histone acetyltransferases, you have histone methyltransferases, and then you have the protein arginine. So it's going to be an arginine residue methyltransferases. Those are three main player writers in the epigenomic signature. So when you do that, that alters now covalently the chromatin. Now that alteration gives that locus of the genome a new way of being expressed. And the way that it's expressed is through reading of that epigenomic superimposition on the genome. And so the reading is going to be done by one of the main players we're going to discuss the rest of this talk. And these are proteins called proteins that have bromodomains. You also have proteins that have chromodomains and tutor-domains. We're going to discuss just the bromodomain containing proteins, okay? Now, bromodomain, for those of you that have a chemistry background, know that it has nothing to do with bromine, okay? The first time I saw that in the literature, I thought, why is it talking about bromodomains? Is it binding bromine or something, or the element? Not at all. Actually, that's a corruption of a Drosophila molecular genetics term for a particular mutant phenotype in that fly called the Brahma mutation. So how Brahma, with a capital B, turned into bromo is anybody's guess. But bromodomains used to be called bromodomains in Drosophila. Nothing more than a structural motif that is associated with the mutation. Okay? So that's what bromodomain. Just I don't want you to ever think it has anything to do with bromine. Anyway, that's going to read this new epigenome signature. It's going to read these acetylated and methylated portions of the genome. And when it does that, it's going to alter transcription. It's going to alter particularly chromatin remodeling at those loci that have these epigenomic alterations, okay? Because it's what's going to be read by the reader. Now, this is a reversible phenomenon. Even though you can indeed inherit epigenome, can do it, the epigenome can be erased, okay? It can be erased somatically, okay, that is before mitosis, or it can even be erased before meiosis. So the germline can often not receive the epigenome, and only the somatic tissue does. It depends on the penetrance of the epigenomic signature, okay? But the erasers are nothing more than deacetylases, okay? And proteins that remove methyl groups from the lysine residues, so lysine demethylases or KDMs. So these are going to be the enzymes that are going to erase the epigenomic signature, and then the readers won't work anymore, and you're back to having basically a naked genome, one that is not altered by the superimposition of the epigenetic signature, okay? So I want you to, this is a recollection. It's also probably one level deeper at the biochemical level about what we mean when we talk about epigenetics. I'm talking now about the molecular architecture of it. Now remember, all I want to say about the global thing about epigenetics is that it alters gene expression from a basal genome without changing the sequence. That's what epigenomics is about. So the bromodomain, in associating with another subunit domain called the extraterminal protein, are proteins in a family called BET, or BETs. Now, BETs are a family of epigenetic readers. They're going to have the bromodomain, and they're going to have this extraterminal domain. So they're called BETs. This diagram came from a Nature Reviews drug discovery paper some 3 years ago. Okay, now one of the core papers I want to talk about today is from my favorite journal, Journal of Biological Chemistry. The paper was published almost 2 years ago, about a year and a half ago. It's going to talk about 4 main BETs. Those are the 4 main ones. BRD2, 3, 4 is going to be the main player in this talk, and then there's BRDT. Each of those have 2 bromodomains and an extraterminal domain. Okay, so they are BETs. And what they do, what their business is to bind those acetylated lysines that were left behind by the writer, the epigenomic writers, right? So the acetyltransferases, right? So you have acetylated lysines on the histone residues. They're being read by the BETs, okay? And because they have this extraterminal domain, this whole operation is also going to mediate a combinatorial protein-protein intramolecular interaction between those domains associated directly with acetylated lysines and the histone tail, and then because of that, three-dimensional in space and time interaction with transcription factors, thus enhancing or at least modifying gene expression that are now endowed with that acetylated lysine being read by the bromodomains. Okay. So BRD4 in particular, let's focus in on that one, regulates gene transcription. It's critical for cell cycle progression and for cell division. Okay. BRD4 acts as a super enhancer. So when this bromodomain is acting inappropriately, it can super enhance the expression of oncogenes, oncogene like MYC, MYC. So this has been recognized for a long time in lots of cancers, not just triple negative breast cancer, but also, for example, in some of the aggressive forms of ovarian cancer. Okay, so because of this, there have been pharmaceutical interventions and drug discovery to find inhibitors of this bromodomain protein. So BRD4 inhibitory drugs can selectively suppress the oncogenic drivers. So this has to do with, because c-MYC is going to push the cells into tumorigenesis. So if you can downplay the super enhancement because of the reading of the epigenome, that is the acetylated lysines that were left behind by the writers of the epigenomic signature, then if you inhibit that whole system, you should decrease the amount of oncogene expression or silence it. And that's one of the things that people believe the inhibitors do. As they're going to find out, though, they also do something else, which is even more interesting, and because it's more complicated, it's both interesting and it's going to take the rest of the talk to explain. So BET inhibitors compete with acetylated lysines, okay? So that's what the drug does. In fact, I'll show you one of the BET inhibitors, in fact, the one used in this paper, and it looks a lot like a modified nucleotide. So when you are modifying DNA protein interactions, which is what we're doing here basically, the compounds that are discovered and that are brought up to the repertoire, the low molecular mass compounds that are used as inhibitors for this kind of inappropriate gene expression, are often mimics of what nucleotides look like. And so that's what these are going to, they're going to resemble chemically nucleotides in their structure, but they're going to be modified in such a way that they have potent selectivity. Okay, so the inhibitors compete with the acetylated lysines, binding to the bromodomain, and basically take that bromodomain out of activity. Okay, so you no longer can recognize the acetylated lysines. And by the way, when you acetylate histones in general, you open up that DNA. When I say open up, in the process of structurally unraveling the double-stranded DNA from the cohering histone, because the acetate's a water-soluble molecule, just two-carbon compound, acetate, right? Because it's water-soluble in an otherwise very hydrophobic region of the cell, which is where the chromatin is packed in the nucleus, a very hydrophobic region. You acetylate, you're going to allow that to open up to all of the potential proteins and polypeptides that are in the nucleus. Thus, that is associated with chromatin remodeling, opening up of the nucleosomal DNA and allowing for things like replication, recombination, repair, and transcription. Okay, so simply adding an acetate turns that chromatin into biologically active chromatin. Okay, it goes from heterochromatin to euchromatin for those of you that follow that in genetics. Okay, so obviously it's going to have an efficacy in cancer if we can block the expression of, say, c-MYC, or we can somehow alter tumorigenesis by altering the reading of that acetylation pattern. So there's efficacy with these BETs. I call them BETs because the I stands for inhibitor. So BETs. There's efficacy in cancer, in inflammatory diseases, and also indeed in the far-flung heart failure. Okay, not uncommon to have drugs be multivalent in human pathologies, actually, because all of these players are involved. So you can imagine DNA repair, which is what this is going to lock back into. Remember the Bracha mutations. It's going to be involved in lots of diseases, including those caused by etiologic agents like viruses, fungi, bacteria. Okay. So Betty act by disrupting the super enhancing effect that normally drive expression of key oncogenes like c-Myc. And that then will lead to, because it prevents that proliferatory response and the cell cycle gets arrested, signals come on and that can generate programmed cell death. That is apoptosis. Betty's also, though, work through another system, okay? Besides altering the expression of oncogenic proteins, they also work through a class of kinases we're going to see real soon here. Again, that's another main discovery in this paper, this JBC paper. Okay, here's the basic summary, and I'll show you a little data. Betty's repress the growth of multiple in vitro and in vivo models of triple-negative breast cancer. They do so by inducing two terminal responses. So this is just biology. They cause programmed cell death and they cause cellular aging to quiescence. We call that senescence, okay? So an aged cell is no longer capable of performing mitosis any longer. It's no longer capable of performing a mutation that can let it fall back into mitotic divisions, okay? Unless it's been altered in some other way. And we talked about that as well. When you get a secretory phenotype or a senescence-associated secretory phenotype, then sometimes you can take a senescent cell and put it back into the population of dividing cells. But generally speaking, senescent cells, they're done, they're finished, they're aged, and the next step for them is going to be to go through necrosis. So what's good about this BET-I, it represses this triple-negative breast cancer via two well-known biological consequences of all living things. They either kill themselves via programmed cell death or they die by aging, which is what you want the tumor cell to do. So the activity of the Bettys and TNBC is not dependent upon suppression of the MYC. So unlike what we talked about before, that BET inhibition looked like because it blocked oncogene expression, that's the main reason it can then tear down the cancer. In this system with triple negative breast cancer, according to this paper and many others that I've looked at, that's not the mechanism. The mechanism is something different. Rather, both senescence and apoptosis are obtained through the accumulation of polyploid cells. Polyploid just means having more than one genome. Okay, so polyploidy in a cell doesn't look healthy to the machinery, like the immune surveillance machinery or the apoptotic signaling machinery that detects something like multinucleated cells and determines that those cells are mutated because you have multinucleation, and those are going to be earmarked for programmed cell death. So you start off by getting polyploid cells, and that polyploidy, as it works out, is afforded or occurs via the suppression of two kinases, the Aurora kinases A and B. Okay, kinases, remember, add phosphate to proteins. And so therefore, those proteins are important mediators of mitosis. So if you suppress that, you don't get mitosis. What you get other than that is you start getting, so you get prepared, you get DNA synthesis, you go through the S phase in the cell cycle, but you don't get to the M phase. So it means you make a lot extra DNA, you replicate the genome, so you get polyploidy in one cell, but you don't go through cell division or cytokinesis. Therefore, that looks bad. The cell cycle recognized via checkpoint as being corrupted, and that's why you lead to programmed cell death, right? So that's where the Aurora kinase is suppressing them, is part and parcel of inducing polyploidy and therefore programmed cell death or senescence. Okay. In fact, if you use specific chemical inhibitors of the Aurora kinases, which exist, that you can buy, they phenocopy the effects of the Bettys. Phenocopy just means they act the same way at the a holistic molecular level, ultimately saying that the phenotype of what Bettys do is they cause apoptosis and senescence, and we think we know what they're doing. Remember the bromodomain and all that, incorrectly named for the Brahma of Drosophila. Remember how that works. Well, as it turns out, the exact molecular signature downstream from where the bromodomains are working seems to be associated with the Aurora kinase suppression. And ultimately, you might guess that's going to have something to do with the expression of those genes, right? And bromodomains can have a role in that. So Betty suppresses the expression, that is transcription, of the aurora kinases A and B. It does so through direct loss of the binding of the bromodomain reader. Okay, and that reader is going on aurora kinase promoters, and those promoters have acetylated lysines. Okay, so the whole thing fits together. Now, I told you that this epigenomic alteration is common. It's extremely common, and it happens in real time, and it turns over. So you can get acetylation of a bunch of lysines. You can get bromodomain associated chromatin remodeling, activation, and expression of that region of the chromosome. And then those acetate groups can be removed, right, by deacetylases. And when that happens, you notice it next round of division. This is, in fact, the subtle way that genes are expressed and then turned off, expressed and then turned off during phases of cell growth and cell cycle in all cells. There's an epigenomic programming, and then there's an epigenomic erasure. Okay, and so we didn't understand this. We biochemists only thought that epigenetics was some unusual phenomenon because of the external environment causing these alterations, methylations, acetylations, which generally either block gene expression or activated it. Simple reminder is when you acetylate a histone, you basically tune up expression of genes. But when you methylate cytosines, when you had a methyl group, which is a hydrophobic group, right, CH3, you turn down the expression of those genes. So it's a way of amplifying and then suppressing, right, the expression of a suite of genes very specifically, and never even leaving a fingerprint, because if you remove the acetates and remove the methyl groups, the only time you would have known that ever happens if you catch it in the act, right? So that's why it went a long time unnoticed, because we didn't have the tools to catch it in the act. Now we do. So beta suppress the expression of these aurora kinases through direct loss of that binding, and this is followed by the generation of these multinucleated cells I just told you, the polyploidy, and the fate of that associated cell line in the tumor lineage then will lead them down the dark road of death, which is what you want tumor cells to take, either programmed cell death, which is nice and neat and clean. They don't signal any other things happening, so they don't cause an inflammatory response, which would be great in lots of cancers, because sometimes you overshoot the mark when you get an inflammatory response. Or they go into senescence, and as long as they don't go through a secretory pathway for senescence, that's also an excellent way to end the life of the tumor. Here's some data. So Betty induction of apoptosis in 4 cell lines. You got 468, 143, 231, and 549 down here. This is jacking up the amount of the Betty, the compound. I told you I'd show you a structure of it. There's the BET inhibitor JQ1, and it looks like this. Now, I know it's got the sulfur atom, but it does have a lot of nucleotide structural similarity. Okay, it also has this chloride, which is interesting because that alters its solubility. Okay, and also this group up here will allow for this compound to have some amphipathic nature that is working both in hydrophobic and hydrophilic systems, and all good drugs are able to do that. At any rate, what you're seeing here, these are 4 cell lines from 4 women who have triple negative breast cancer. These are cell lines from people. And you can see that as you increase the amount of the Betty, right, the drug that blocks the bromodomain binding to the acetylysines, you increase apoptosis. But you increase apoptosis according to concentration, but also according to cell lineage. Okay, so this JQ1, which is a Betty, it's a kind of compound, remember, that is going to affect the amount of acetyllysine that gets recognized and therefore gene expressed. Okay, so it's going to alter that. So you can see that there's a cell line associated alteration. Okay, cell line associated alteration. So that's what we're telling you here. So JQ1 increased the number of apoptotic cells in all 4 cell lines, but 2 of the lines, 468 and 549, had a more pronounced apoptotic response. So that means that there is a difference between the 4 cell lines, which are all from triple negative breast cancer cell lines, in their reactivity to the bromodomain, extraterminal domain Betty protein inhibition. Okay, that's the important point here. Now, tumor volume, which is another measure of decreasing tumor growth, was suppressed in both Claudin low and in basal. Those are the two subtypes I told you we would mention, triple negative breast cancers. After treatment with the Betty, even though the apoptotic response was more aggressive in the 468s versus the 231 cell lines. Okay, so remember that in the last slide, we saw that the 468 showed a more aggressive apoptotic response. Yet, it doesn't appear that this differentiates according to whether or not these are basal or Claudin low. So, in a way, that's a good thing because these drugs, this Betty, will be able to block those two subtypes of triple negative breast cancer. Okay, so you won't get a selectivity where one is spared and one is inhibited from continuing to make cell mass that is increased in apoptosis. And you can see the relative tumor volume is decreased in both of them, even though they're disparate in the subtype. So same thing shown here. You have so-called senescent types and apoptotic types. The senescent types are 231 and 143. And the apoptotic types are 468 and 549. That means that these cells tend to become senescent. These go directly to apoptosis. And you can see that when you add the drug, you still get apoptosis with a baseline of senescence. You also get apoptosis with a baseline of apoptotic cell lineages. But you get the phenocopy, you get the same effect when you add the drug that inhibits those aurora kinases. That's what AZD is. You add the aurora kinase, you also get apoptosis in all the cell lineages. Nice boost. So it's phenocopying, you see, the JQ1, which is the Betty. Okay? In fact, it's giving you a little bit of a higher response than the Betty. So that's probably because it's downstream. So I want to bring up something now that throws kind of like the major curve in the understanding that I want you to come with me on as we're going down this road. Now, this comes from a different paper, very recent paper. Came out 26th of July, just a 3 months ago this year. This protein talks about repression of BET activity, so Betty, okay? And what that does is sensitize homologous recombination proficient cancers to PARP inhibition. Now, PARP, okay, follow along here, is polyadenosine diphosphate ribose polymerase. Now, that enzymatic activity is involved in the early stages of homologous recombination repair of damaged DNA. Okay, so when you inhibit PARP, you prevent the early phase of HR, that's homologous recombinatorial mediated DNA damage repair. Okay, so DDR is going to be decreased when PARP inhibitors are used. Now, PARP inhibitors are a new type of drug that's out there for all forms of cancer, not just for triple negative breast cancer. Okay, you find it in non-small cell lung carcinoma. You find it in hepatocellular carcinoma. You even see PARP inhibitors being used in renal carcinoma and in glioblastoma. Okay, now what this paper showed is that if you add another drug, a BET inhibitor, in fact, the same one we saw in our JBC paper, JQ1, remember it's a BET inhibitor, you get an enhancement of this alteration associated with PARP. So this is happening in primary and acquired homologous recombination proficient tumors. And all of that is facilitated by adding Betty via the inhibition now, I'm adding all of them together right now, and I'll elaborate, of BRCA. Now, what that means is, remember, we went way back, many eons and moons ago, the beginning slides. I told you that Bracha1 mutations are typically linked with triple negative breast cancer. That's true. And I told you that because you get a mutation in BRCA1, you don't get good DNA damage repair. That's true. Poor DNA damage repair means that cells can carry mutations and yet can go through pass all the checkpoints, continue to replicate and go through mitosis. And in so doing, replicate mutant cell lineages. And of course, that is a hallmark of the beginning of oncogenic events leading to tumor growth, tumor division. Okay. Now, what often is misunderstood here is that the mutation that causes the primary cancer is also a mutation that if you push it over the mark, and if you make it completely irreparable and it doesn't work at all, it will kill the tumor cells. Okay? So imagine something that causes the tumor cells to get sick, healthy cells to get sick, so they start dividing autonomously and turn into tumor. Now imagine that same mechanism gets jacked up so that not only do you get more and more and more of this mutation being afforded, but because of that, okay, it actually alters the expression of the initial player that induced the mutation. So if the BRCA1 has been selected for in the tumor cell lineages, which it would be, because those are the ones that are going to survive, you see, and now what you do is you knock down the activity of the BRCA1, okay? That's what this is happening. You're getting a relative intensity decrease by adding the Betty. You're getting less BRCA1. Now you have no way to control DNA damage. There's no homologous recombination mediated repair or any of the other. There's now non-homologous end joining repair. There's no quick way to remedy this problem with further mutations. And so now the tumor cells have gone way out on the limb, expressing only the BRCA1. They're expecting the BRCA1 to carry out some kind of DNA repair so that you don't get so much damage that you have no genome integrity and the cells won't even be able to grow. Certainly not divide. That's what this does, pushes them over the edge. So that's what PARP-Is and Bettys do. Working in combination in this paper that was in Science Translational Medicine, one of my favorite new journals, knock down BRCA1. This to me looks like a real good possibility of multivalent, multi-drug approach to go after triple negative breast cancer. This looks like this could work. Now, there are some caveats to that, obviously. First of all, you have to have this proliferation of the BRCA1 mutation. You have to be poised just correctly so that if you start decreasing overall DNA repair, that you're not hitting other host cells. You have to target these drugs, obviously, to tumors. That's a big feature, right? All right, so let's conclude and make some induction. Inhibition of the BET lowers the expression of the aurora kinases. That was the JBC paper. Those are normally necessary for cell division. So because you don't get cell division, the cell cycle proceeds through the S phase, so you get polyploidy, okay? That's going to lead to growth arrest. So the cells respond to this polyploidy by either undergoing senescence or ultimate aging or apoptosis. Okay, so in other words, there's a recognition of something wrong with these cells and they die. Now, another way, of course, is that T cells can recognize them. Cytotoxic T cells, for example. Now, what I didn't talk about, didn't show you any data, is that Bettys have been further examined, and they also reduce in animal models for triple negative breast cancer liver metastasis after primary breast cancer. Okay, so that's looking good, right? So these drugs tend to work also in controlling metastasis. And that's a major problem. Remember, that's a major problem with triple negative breast cancer. That's why it's so aggressive, because you get a lot of metastasis, for example, to the liver. So aurora kinases A and B expression may serve as biomarkers, according to that JBC paper, because certainly they're involved in this whole pedigree leading down to tumorigenesis. But they're also maybe very good targets, because we saw in that phenocopy effect of the aurora kinase inhibitors. This is another whole new drug line that may come out now because of this paper. Now, let's get back to talking about some more clinical aspects of it. The main reason this caught my attention because this disease is so devastating, and I would really like to see more research, more money thrown at this so we can attack this terrible high mortality breast cancer in women. Here are 6 papers I looked at. Here's the American Journal of Pathology. This is one of the first ones I saw. Talks about the fact that African American women, both premenopausal and postmenopausal, remember the earlier age onset, are more likely than non-Hispanic European American women, that would be like Caucasian women, according to this paper, to be obese and to be diagnosed with triple-negative breast cancer. So this paper was trying to determine if there was a distinction between a racial distinction or a distinction of hypermetabolic activity, such as obesity. So it's trying to break those two things apart. And the paper really wasn't able to do that. The paper wasn't able to say, why do African-American women get triple-negative breast cancer more often than non-African American women? And they weren't able to determine whether or not obesity was the main player, although that's often been associated with triple-negative breast cancer. So let's continue on. Second paper we can discuss. This came out of Cancer Epidemiological Biomarkers and Prevention in 2008. Triple-negative breast cancers occur within a younger white female population, so age is common, earlier age, premenopausal, for both African-American and white women. And this one was in association with obesity. So this paper was 8 years earlier than this one. So that's why they were saying maybe obesity is the linkage, right? But there was no relationship. In the third paper I found, this is in the Asian Pacific Journal of Cancer Prevention, 2013, more recent, no relationship between obesity and TNBC, triple-negative breast cancer, in Turkish women. Now this, once again, though, this is a different breeding population, right? So it has, again, something to do possibly with where we find triple-negative breast cancer worldwide, okay? And the reason I say that is, let's continue on. This paper came out in 2015 in Breast Disease. Triple-negative breast cancer subtype in Algerian women was higher compared with Caucasian women of European ancestry living in Algeria. So once again, it looks like there could be some racial effect, right? That Algerian women who are non-Caucasian tend to have higher incidence of triple-negative breast cancer. Now I should mention, and I did at the beginning, but I will say it again, triple-negative breast cancer only accounts for a maximum of 20% of all breast cancers. So breast cancer in general, that is the estrogen receptor and the progesterone receptor and the HER2 receptor type breast cancer, is actually more common in Caucasian women. Okay. Now, the way those numbers play out, it looks like the triple-negative breast cancer is more common in Black women, but it's not necessarily, you see, associated with racial differences. It simply means, it could mean, that African American women get less of the estrogen, progesterone, HER2 subtype of breast cancer, the more common one. See, it's still trying to work this out, right? Let's go on. Next paper we're going to talk about comes from a journal called Springer Plus, 2016. Central obesity, okay, especially with a high BMI, right, was an independent prognostic factor for triple-negative breast cancer in Chinese women. Okay, again, a different breeding population, right? So now we're looking at Asian populations, we're looking at African-American populations, we're looking at Caucasian populations, either European or non-European. We're also looking at subtypes of African populations, such as in Algeria, and we're even looking in the Middle East in Turkish populations. And we're seeing differences in the triple-negative breast cancer, and it looks like sometimes associated with obesity and sometimes not. It certainly seems to be associated with geography, right? And therefore could have something to do with genomes. Last paper, very recent, breast cancer research, just came out just very, very recently. Breastfeeding among African American women decreases triple-negative breast cancer. Now, this was a study done, again, with multiparous women who have already had children. And the question is asked often in the hormone-sensitive breast cancers, and probably most people have heard this, that women who have had children that have breastfed those children tend to get less breast cancer. Okay, and it's particularly the main form of breast cancer, the hormone-sensitive types. As it turns out, at least with this one study, and this is brand new, so it's going to have to be replicated, of course, looking just at one population of women, African American women, those that did breastfeeding versus those that did not tended to have lower triple negative breast cancer compared to their counterparts in the same cohort population. So that's interesting too. So let's put this all together into a dialectic, and we're going to use a paper that was published in Cancer Biological Medicine in 2015. So let's put this together. Triple negative breast cancer is more commonly diagnosed in younger and premenopausal women. It's highly prevalent in African American women as compared to other forms of breast cancer. Breast cancer is the second leading cause of death overall in women after lung cancer, and it is the most common cancer among women worldwide. Just it's not the one that causes the highest mortality, that would be the lung. So the highest overall cancer in women, again, it's because of it's a ductal carcinoma. 23% of all cancer cases are going to be breast cancers, all breast cancers. You get about 1 million new cases of breast cancer every year. So terrible disease. About 170,000 of those, you see, up to about 20%, are your triple negative estrogen receptor, progesterone receptor, HER2 receptor minuses phenotype. Okay, triple negative. Now, besides the drugs we talked about, major medical procedures are also used for the triple negative. What are they? Surgery. Both mastectomy and lumpectomy are used for triple negative breast cancers in combination with radiation therapy and chemotherapy. They've all been commonly done, but even with surgery, radiation therapy, and conventional chemotherapy, prognosis is still very poor when you have triple negative breast cancer. Main reason, I think, is because we have really good drugs to block things like estrogen binding to its receptor and progesterone binding to its receptor. You see, like tamoxifen for the estrogen receptor. Of course, you do get resistance, though. You do get drug resistance. And there are other problems with the whole estrogen pathway. However, we don't have that kind of magic bullet for the triple negative breast cancer. So it could be the reason you get bad prognosis. We don't have one really magic drug, which is very good at knocking out triple negative. Maybe we're starting to find it, though. Maybe with a combination of the BETIs, right? The bromodomain, extraterminal domain inhibitor compounds, like we looked at here. Maybe in combination with PARP inhibition, right? And therefore decreasing the BRCA expression. And once the BRCA population is moved on so that the mutant population can be diminished and you get senescence or programmed cell death, maybe that's a new way to go with this disease. So what I want to say in conclusion here is that immunotherapy, okay, and we talked about that in a couple of previous talks, and this epigenetic therapy we just looked at are becoming important in current clinical trials, okay? So women are starting to enroll in clinical trials looking at both immunotherapy, that's checkpoint type PDL and PD inhibition in T cells so that you get an activated T cell. T cell then, sometimes T cells can kill tumor cells. It goes that route. So immunotherapy is affecting the T cell population, not directly the tumor population. Epigenetic therapy we just talked about. Now, that's becoming more prominent. And what I would like to say is the etiology and the occurrence within specific populations of women, according to age, race, comorbidity, that is other disabilities, and perhaps just weight or mass, body mass index, need to be more thoroughly studied. So we need to get more money from Big Pharma, more money from the NIH to look at triple negative breast cancer and try to get that down, because that's a very aggressive, very dangerous disease. It's killing a lot of women. We want to get rid of that. Now, I want to just remind you some key features that we discussed. Acetylation signaling has to do with an acetate molecule on a lysine residue. These remember these bromodomain containing proteins like the birds read that. Remember that the acetate is added by histone acetyltransferase. That's a writer, and the eraser is going to be a deacetylase. Okay. In fact, the HDACs, there's a whole family of them I've talked about in other VerEvMed talks. They're called sirtuins. Proteins called sirtuins or HDACs. That's another whole interest in cancer biology, sirtuins. Now, when you have this whole epigenetic phenomenon going on here. That's what this box represents. You can get DNA damage response because these genes can be controlled. The expression of these genes can be controlled via this acetylation pattern and can turn on the damage response. For example, the normal processing of the BRCA pathway. Or if you get mutations in the epigenomic patterning, you can get mutations. You get alterations of expression activity, that is, genes are inappropriately expressed. That's going to lead to genome and epigenomic instability and cancer. Therefore, pushing that out of the danger zone, the cancer zone, back into fixing DNA damage, once again, is another strategy, a backdoor strategy to looking at this kind of cancer. So anti-cancer therapies include things like removing acetates, okay, and also adding acetate. See, so you can attack it from both ways. You remove acetate, you get rid of the signature. Maybe you need to do that when you have a mutation in this system, say in the bromodomain protein itself, so you don't even want to go down this pathway, or you find drugs that affect removal of the acetate, right? So that you never have added it, so you don't make that molecular signature. So you can see there's a lot of valencies to go after here. Sorry for this last slide. I know it's terrible, but I wanted to show you that this compound we've been looking at, this came from a paper published in Cell Reports in 2016, about a year and 3 months ago. In JQ1 sensitive, the JQ1 blocks, this is tyrosine kinase activity, okay? And it induces apoptosis. Now, this is ovarian cancer, okay? See, we got some players here, c-MYC, so you're shutting off c-MYC because you blocked off this receptor tyrosine kinase, and you did so because this is sensitive to these bromodomain inhibitors. That's a bromodomain mediated response, right? So you don't get ovarian cancer. But in the resistant ones, what happens is you get the autonomous receptor tyrosine kinase activity, and you run through this pathway, and what you do get is c-MYC activity, growth, and then therefore ovarian cancer. So what this paper is telling us, this is ovarian cancer, not breast cancer, is look out, because when you start coming in with new drugs, you're going to get resistant phenotypes. So maybe that PARP inhibitor along with the BET inhibitor will decrease the phenomenology of this quick reception, which ultimately mediates resistance, right? So that's why people look at multiple drugs. Okay, we're finished with this talk today. Thank you very much for your, I'm sure, undivided attention. Again, of course, this is beautiful Canyonlands in Utah. My name is Dan Guerra. I'm VerEvMed's Chief Scientific Officer, and I'm the person that delivers these videos to you. There's my email. You can address me specifically, and we can talk about any of the talks I've presented out on YouTube, as well as what we do at VerEvMed. We'd love to have you as a client. Here's the VerEvMed website right here. There's the email directly to the VerEvMed, which we will also receive. Remember what VerEvMed is. It's our company, and what we do is we are scientists verifying published evidence in medical biosciences. So we're here to consult with people, lay people, and including health professionals on medical biosciences. And what I just did there is give you sort of an example of how we might do that. All right. So again, thanks a lot for paying attention. I hope that you have a pleasant weekend, and we will see you soon. Bye for now.